Here is some picture of our members in group 1. I am a leader, and Mayan is designer, and Minh Anh, Kiều My, Kim Chi, also Di Đạt, a designer. Now let's start. In our presentation, we divide it into 5 parts. The first one is the overview about DNA. We will answer the questions when and why is DNA discovered. The second one is the definition of DNA. In this part, we will let you know what is DNA. And the third one, is why DNA important. The fourth and the fifth are more important than another part. It is DNA structure and the function of DNA. Now, let's move on our presentation. First, I will let you know about the overview about DNA. As we can see, this is the picture describe about DNA. In 1953, James Watson and Francis Crick introduced an elegant double helical model for the structure of deoxyribonucleic acid, or known as DNA. The substance of inheritance is the most celebrated molecule of our time. Here, the tree information is encoded in the DNA and reproduced in all cells of the body. DNA stands for deoxyribonucleic acid. This DNA program directs the development of biochemical, anatomical, physiological, and to some extent, behavioral traits. It is a type of nucleic acid, one of 4 major types of macromolecules, which are essential for life, along with proteins, lipids, and polysaccharides. And it is the end of part 1. Now we will move to part 2, which will be said by Minh Anh.
Thank you for the first information that our leader provides. Hello, I'm Minh Anh. To my turns, I plan to say a few words about the definition of DNA. DNA, which is a molecule that contains the instructions an organism needs to develop, live, and reproduce. These instructions are found inside every cell and are passed down from parents to their offspring. To carry out these functions, DNA sequence must be converted into a message that can be used to produce proteins, which are the complex molecules that do most of the work in our bodies. Each DNA sequence that also contains instructions to make a protein is known as a chain. The size of a chain may vary greatly, ranging from about 1,000 bases to 1 million bases in humans. A single DNA molecule usually contains many chains. Well, how do you feel after being heard about this? To me, I'm so surprised at the number of the base in only one chain. 2 pictures you see on the screen express simply the division from one original DNA to 2 new ones. Now let's move to the third part of this presentation belonging to Mayan.
Thank you, Minh Anh, and hi everybody. My name is Mayan, and welcome to my presentation. In my part, I will explain to you why is DNA important. DNA is important because it holds all of the genetic information. This information is needed for your development and survival, and it's also be able to be passed along to the next generation. It also influences your taste, ranging from what you look like to the food you like, with lots of things. DNA gives you how different you are from someone else. It might seem like everyone's DNA should be very different from one another. On average, you share around 99.5% of your DNA with someone you are not related to. A big part of what makes you unique is found in that 0.5% of your DNA, and even though we're overall more alike than different, everyone's DNA tells a different story about who their relatives are and where they are from. What story does your DNA tell? And although my presentation is short, but it is also the end of my part. Let's move to next part of Qme.
Hi class, I'm Kyumi, and after my aunt's performance, I'm now here to continue our presentation. My talk has the subject of DNA structure. In this part, we are going to find out the components that create DNA and how DNA actives. First, we come to the nucleotide, which serve the monomeric units of DNA. Each nucleotide is composed of three components. The first one is a phosphate group. The second is a pentyl sugar that contains 5 carbon sugar, and the last one is nitrogenous base. Besides those three subunit molecules, there are also 4 types of nucleotides: adenine, thymine, guanine, and cytosine. The types of nucleotides are only different nitrogen base. People call nucleotides after the names of nitrogenous base. As you can see, the photo below can give you all better understand about what I just said. And this leads me to my next point is the ways nitrogen pair together. DNA molecule consists of two polynucleotide chains linked by hydrogen bonding between nitrogenous base of nucleotides follows the principle of complementarity. Adenine is bonded to thymine by 2 hydrogen bonds. Guanine is bonded to cytosine by 3 hydrogen bonds, and so DNA molecule is both stable and flexible. Two polynucleotide chains both link together and twisted around an axis. Take a look at the picture on my screen. It is easy to see that nucleotides are attached together to form two long strands that spiral to create a structure called a double helix looking like a ladder. Imagine forming a double helix like a spiral staircase. The handrails are sugar molecules and phosphate groups. The ladder is nitrogenous base. The phosphate and sugar molecules would be the sides, and the base pair would be the rungs. Speaking clearly, the hydrogen base on one strand pair with the base on another strand. Adenine pairs with thymine. Guanine pairs with cytosine by hydrogen bonds. Therefore, knowing the base on one strand, we can easily figure out the sequence of base on the other strand. Continuously, the DNA helix splits down the middle and becomes two single strands. They serve as templates for building two new ones, still according to hydrogen bonds rule. Well, that's all I have to say about. But it's not the end of DNA structure. To discover more interesting things about the structure of DNA, we will move to the next part with Soul by Kim Chi.
Thanks for giving me performance. And hello everyone, I'm Kimchi, and now I will follow up on Kimi presentation. I will tell you about set determining chromosome. Nucleic DNA is made of 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes, for a total of 46 individual chromosomes. A chromosome is a discrete set of genetic information with one chromosome of a chromosome pair inherited from your mother and one chromosome inherited from your father. As we see in the picture, there are two types of DNA in the nucleus: autosomal DNA, Y-sex chromosome DNA. Strictly speaking, only chromosomal DNA is unique to each individual, which makes our DNA a powerful tool for DNA identification. Chromosomal DNA testing typically use specific, well-defined loci. We can also call it a loci, far ruled out 22 pairs of autosomes and sex determining chromosome S and Y. And on the screen now, as the picture, they show us about chromosomal in male and female. As we see, the two pictures are almost alike, but there is a different one is the last chromosome. It is the sex determining chromosome. In female chromosome, it is the S DNA, while Y DNA is the sex chromosome of the male. And the next, also the last point I tell you in my presentation is that the difference between two types of DNA structure between eukaryotic cell and prokaryotic cell. The picture on my left hand discusses about eukaryotic ones. In eukaryotic cell, the DNA molecule usually have a linear structure, and the picture on my right hand mentions to the prokaryotic cell. In prokaryotic cell, the DNA molecule is usually circular in structure. And it is the end of my presentation. Now let's come to the function of DNA, and Duy will be the ones who perform it.
Thanks for Kimchi presentation. Hi everyone, I am glad for this opportunity to introduce myself. My name is Das, and I will continue a part of Kimchi presentation. Now I will show you to the function of DNA. The function of DNA is tied to its structure, and as discussed above, DNA is a database for the life on the current planet. DNA is a repository of all information about movement, activity, and reproduction of organisms, which are the complex molecules that do most of the work in our bodies. It's basically your biological rule and very useful to review to understand its functions. DNA is responsible for carrying, preserving, and transmitting genetic information. Genetic information is stored in the DNA molecules in the form of the number, composition, and order of the nucleotides. The sequence of nucleotides on DNA is responsible for coding for the sequence of amino acids in the polypeptide protein chain. Proteins make up cells and thus determine the characteristics of an organism. Thus, the information on DNA can determine on the characteristics of an organism. And the next topic will be presented by Bình.
Hello everyone, and I am Bình again. You must have been very tired for listening until now, but don't worry, we are nearly finished. Now, let's continue the content given by that. The genetic information on the DNA molecule is preserved very tightly. Errors in the DNA molecule are all repaired by the system of repair enzymes in a cell. genetic information on DNA chains is also transferred from DNA to RNA and into protein through the processes of transcription and translation. Apart from storing genetic information, DNA is involved in 7 small parts. The first one, replication process, means transferring the genetic information from one cell to its daughters and from one generation to the next, and equal distribution of DNA during the cell's division. The second one is mutation. It means the changes which occur in the DNA. And the 5 other part is sequences, transcription, cellular metabolism, DNA fingerprinting, and gene therapy, respectively. And it is the diagram of the transcription of the IDN. And this diagram is also the end of our presentation. We are group 1, so we make many mistakes. I hope everyone will forgive it. And this the chart is illustrate our contribution to the act of the presenting. Thank you for listening until now.
Thank you, group 1. And now, group 3, present the structure and function of DNA. Group 3.
Dạ, để em chia sẻ thầy.
Okay.
Từ từ.
Now, begin, start.
Okay. Good evening, class. Welcome to our group 3's presentation, PowerPoint, about DNA structure and function. We from Bình High School for the Gifted, class 10E9, and the teacher is Mr. Đỗ Vĩnh Lộc. Lesson 6, Nucleic acid, DNA structure and function. I'm the leader of this group. My name is Nguyễn Thị Yến Linh. Our group has 6 members. They are Hoàng Trung Kiên, Nguyễn Lê Xuân Mai, Phạm Nguyên Linh, Nguyễn Trương Phương Linh, Phạm Vũ Hà Linh, and me. Let's talk with us. In this topic, we have 5 parts. The first part is the introduction. The second is the structure. The third is the function. The next is the in conclusion. And the finally is the comparison between DNA and RNA. The first part, introduction. At this part, Miss Mai will present it to you. Let's listen to her.
Yes, I'm Mai. Part 1, introduction. DNA is the information molecule. It's the instruction for making other large molecules called proteins. The instructions are stored inside of your cell, distributed among 46 long structures called chromosomes. The chromosomes are made up of thousands of subtle segments of DNA, called genes.
Each gene starts the direction of making protein fragments. All proteins are multiple specific proteins that have partial structure of DNA. This link will continue our presentation.
Thank you, Mai. DNA is a biological macromolecule polymer with a polymolecular structure made up of many monomers. Each monomer is a nucleotide. Each nucleotide is made up of a nitrogenous base, a pentose sugar, and a phosphate. DNA has 4 types of nucleotide: adenine, thymine, cytosine, and guanine. These types of nucleotide differ in their nuclear base, nitrogenous base composition. So their name will based on the nuclear base they carry, in which A and G are purines, lysosine, and T and H are pyrimides, smaller type. The association of nucleotides together in a certain direction forms a chain called a polynucleotide. Each DNA molecule consists of 2 polynucleotide chains. In aqueous medium, the 2 polynucleotide chains of a DNA molecule are linked together by hydrogen bonds. This bond is formed between the nuclear base of the nucleotides overlap each other on the 2 chains. And the next part of structure of DNA, Wang Lin will continue. Let's listen to him.
I will continue, Ms. Ling. Due to the chemical structure of nucleobase, this bond is very specific. Hydrogen bonds only form between 2 certain types of nucleobase, namely A and T. 2 hydrogen bonds, X and G, 3 hydrogen bonds. In fact, is a link between a purine and a pyrimidine, so the relative distance between the 2 polynucleotide chains is pointwise stable and flexible. The DNA molecule is a double helix because the 2 polynucleotide chains of the molecule are not only linked together by hydrogen bonds, but they are also twisted around an imaginary axis from left to right containing sugar, deoxyribose, and phosphate. DNA is encoded with a triplet code containing 3 of the 4 nucleotides A, T, G, and C. Next part is the function of DNA. This is the part of Miss Phương Linh. Listen to her.
Good evening, everyone. Now I will introduce you about the function of the DNA. DNA is responsible for carrying and storing genetic information, which is stored in the DNA molecule in terms of the number of components and the order of nucleotides. The genetic information on the DNA molecules is very tightly preserved. Errors in the DNA molecules are only corrected by the enzyme system in the cell. The information on DNA is passed from cell to cell, inferred by DNA replication of the cell division. The genetic information contains data on the structure and characteristics of all the proteins present in the organism, thus contributing to the regulation of the characteristics of the organism. To continue, Chung King will summarize the coding content of the structure and the function of DNA.
Thank you, Phương Linh. To summarize our presentation, DNA is a biological macromolecule with a polymolecular structure made up of many monomers, and each monomer is a nucleotide. DNA is made up of 2 polynucleotide chains linked together according to the principle of complementarity. A is linked to T by 2 hydrogen bonds, and G is linked to X by 3 hydrogen bonds. And the function of DNA is to carry, preserve, and store genetic information. And DNA is quite similar to RNA, so to avoid causing confusion with DNA and RNA, Helen will tell us about the similarities and differences between them.
Thank Mr. Kyun. First of all, I will say the similarity between DNA and RNA. Both DNA and RNA store genetic information. Moreover, both DNA and RNA consist of sugar, nitrogenous base, and a phosphate. Finally, on both molecules, guanine and cytosine pair with each other. Next, I will say the difference between them. DNA uses the sugar deoxyribose, while RNA uses the sugar ribose. Secondly, DNA uses the base adenine, thymine, guanine, cytosine, while RNA uses the base adenine, uracil, guanine, cytosine. Finally, DNA stores and transfers genetic information, while RNA acts as a messenger between DNA and ribosome to make amino acids and proteins.
And moreover, we have made a waste board. This board assigns tasks according to the percentage of each group member. Everyone can refer it, and maybe this is the first time to giving a presentation, so our pronunciation is still lacking. Hope everyone can understand for us. And